So, good morning to all of you. This is Rajat Pal this end from the Department of Microbiology and Biotechnology, Sister Nivedita University. On behalf of this department, this beautiful department, incredible department, I welcome you all for this 5 days online introductory course to microbiology and biotechnology. With me, there are Dr. Shomshubra Thakur Chaudhary, Dr. Fatima Kalkatawala, Dr. Atrey Ghosh, and Dr. Milan Banik is there with us. And the all this program and the concept and the everything is under the guidance and the blessings of Professor Jubojyoti Chattopadhyay, Honorable Vice Chancellor, Sister Nivedita University. He has kindly agreed to deliver the welcome address for this session. Sir, hello, sir. Hello, sir. Sir, are you there? Hello, DJC, sir, are you there? Hello, DJC, sir, are you there? Maybe sir has got disconnected. Fatima or SCC, can you please contact once, sir?
Okay, sure, Rajat, I'm calling.
Yes, yes. You just see whether sir has joined or not.
Yes, I have
Yes,
joined. I'm
sir.
waiting.
Oh, sorry, sorry, sir. So, shall we start now? And would you like to give the welcome address now, or you will join us later on, or you will give the welcome address after some time, sir? That is just our query to
I
you.
shall request you to start the meeting and then I shall
Okay,
give
sir.
my,
Okay.
yeah,
Okay. Thank you,
give
sir.
my
Thanks a lot.
address. Yeah.
Thank you very much. Okay, sir. Okay. So, with the permission of our Honorable Vice Chancellor, Professor Dibojiti Chattopadhyay, we are going to start our program. So, at first, on the behalf of this department, I would like to share you some thoughts and some views of our department in the very beginning. Then, Professor Chattopadhyay will give you the welcome address. Um, I am just sharing some presentation to you on our department. Can it be shared? Is it visible to everyone?
Yes, Rajat, that's visible.
Okay. Okay. So we are going to talk about the Department of Microbiology and Biotechnology, Sister Nivedita University, Kolkata. As I told that I'm Dr. Rajat Pal, right now heading the department, and with me there are four incredible faculty members I do have with me. That is Professor Atrey Ghosh, Professor Shomshubra Thakur Chaudhary, Professor Fatima Kalkatawala, and Professor Milan Boni. We five are running this department. Okay. In our department, two courses are actually running simultaneously. One is the biotechnology, the other one is the microbiology. For both, we offer UG and PG courses. Okay, these are some photographs of our well-equipped laboratories. And as I told you that biotechnology and microbiology, both the departments are running simultaneously under the same roof. And for both, we are, offering the postgraduate and undergraduate programs. What will be the future prospects or job prospects for these subjects. The higher studies in abroad is the major focus to our students. We always encourage our students to study more and more and more, and after graduation they should study the post graduation. After post graduation study they will go for the PhD, whether in India or outside the India, any universities. Already we have some foreign collaborations, and our students are going to the different outside countries. for their higher studies as PhD. Okay. Many jobs in hospitals, pharmaceutical and biotechnological companies are open for our students. May I request everyone to mute their mic, please? Yes. So this is the major focus of the job aspects of our department, mainly the hospitals, the pharmaceutical companies, and biotech companies. There, our students can go and have a job over there. R&D is one of the major sector where my students are absorbed. That is the research and development sector of many MNCs, that is multinational companies and national companies also. As you all know that in this pandemic situation, the research has become a very trust area for all the subjects, mainly especially for microbiology and biotechnology. So after few years or within few years, there will be more companies and more opportunities for the research, how we can come to our normal life. Okay, from new normal to old normal. I don't know whether we will be able to go back to our old normal or not, but still the new normal, how much we can accustomed with the new normal, that we have to see. And the other job opportunity is the my favorite actually personally, that is the teaching line in colleges and universities in anywhere in India and outside the India also. Whenever you are getting your most highest degree, that means the PhD degree, after that you are eligible for, or else if you clear the net examination, sorry, net examination, then you can apply for the faculty position in any colleges or universities in India. So these are the main job focuses of the students from our department, and some highlights of the department. As you can see that we are very much vibrant department. Many, webinars and seminars and lectures, talks we generally organize regularly in regular intervals. The students who are from our department, they are already accustomed with that, and the students who are actually coming from the other parts of the India, or I can see that from international participants are also there in our this webinar series or course series. So everyone can see that we generally used to do lots of webinar series, and there's many eminent scientists, eminent doctors and researchers from India as well as from the abroad are giving their lectures. Professor Dhrubajyoti Chattopadhyay, Professor Ashok Ranjan Thakur, they are the big shot in this microbiology and biotechnology field. They actually taught our students. We are blessed that they are also belongs to our department. So these kind of faculties we do have. Hmm. And you can see that there is a group photograph. That is the photograph of a group who went to Okayama University for the collaboration with the sister Nivedita University. Okay, Dr. Shumoli Pine, under guidance of Dr. Shumoli Pine, we can have this collaboration. We are very much fortunate to that. One of our students, you can see her, photograph, the Rupa Rani, she is going to Japan for her PhD. We are quite often publish our research papers. As I told you that in our department, research is a very important thing. All of our faculties are doing research very passionately, and in a regular basis we generally publish our papers, research papers, in international and national journals. You can see my students are a very vibrant student. They achieve so many things, so many certificates, so many medals in the university, many competitions they are taking part. They are achieving a very good marks. They are having very good positions in the academics also, as well as the extracurriculum activities, sports and debates and everything. They are very much involved. We have a library, departmental library. You can see many books are available. There is a central library also, and there is a departmental library. So actually, altogether, it's a very, enthusiastic department. Both academics and the extracurricular activity, everything together. This is actually not only an educational department; it is actually an overall place where the students can enjoy the study. Okay, so this is all about for an introduction of my department. Um, okay, this is it. Now, may I request Dr. Fatima Calcutawala to take over the mic and to start the session. Madam, welcome.
Thank you, Rajata. Rajata, my voice is clearly audible.
Yes. Madam, your voice is totally clearly audible.
Thank you, Rajat sir, for the kind introduction. And hello and welcome all the participants.
One second, Dr. Calcutawala, to interrupt you, I forgot
Yes,
to
yes.
introduce you actually. Okay, I
Okay,
have introduced the department very nicely there. You are a very, important part that I showed. But what you are, that I have to tell in front of my
okay,
students.
sure,
Yes,
Rajat.
I am very much glad and very happy to have Professor Calcutawala with us. She is a NET JRF SPM fellow. And we are very much proud to have Professor Kalkatawala with us. She has done her PhD from NICET Kolkata, and she has done her research mainly in microbiology and virology. And her expertise in teaching is also microbiology and virology. In our department, she is teaching these two subjects, and she is also having her research in this topic. And now you can see that how she will express about the cell and cell organelles and how cell work, everything she will tell you in her lecture. So I invite Professor Kalkatawala to give her lecture, and I cannot say that the stage is yours. I can say that the computer screen is yours, Professor
Okay,
Kalkatawala, now.
thank you,
Okay.
sir. Uh,
Yes.
Rajat, if Vice Chancellor sir is there in the meeting, will he like to give the welcome address right now? Is sir here in the meeting?
I think I cannot see, sir. Uh, Sir, okay, I'm texting sir whenever he will be free, so that he can tell us, so then we can introduce him to the meeting, to the audience, so that he can deliver. Okay, so
Okay,
he
sure.
told
In that
me
case,
to start.
I'll start.
Yes. He told me
Okay,
to start the session, so we can start the session. Whenever he will be free, he will come. When he will join the meeting, then he will deliver her lecture.
sure.
I'm
Thank you.
texting him. Yes,
Rajat.
yes. Hmm.
So, hello and good morning, all the participants. Welcome you all to this introductory course to microbiology and biotechnology. So, what I will be speaking about today is cell, the basic unit of life. So, please have a look at this presentation. I hope it will be very clear to you. I have made very simple slides just to brush up your basics, and then we will end on a slightly complex note. So, I would like to share the screen with you all. Rajat sir, is the screen visible?
Yes, Dr. Kalkatawala, it is clearly visible.
Okay, sure. So, I would like to begin with this presentation, Cell, the Unit of Life. As you all are aware of this fact, that life begins with a single cell. In this figure, we can see a single 200 micrometer human egg cell, which is fertilized with a sperm cell, which is also a single cell. And from the union of this egg with the sperm arises 10 trillion cells of a human body. So that is the beauty of a single cell. It gives rise to an entire organism. Now, if we go back to the basics, we go back to the textbook definitions of a cell. Cell comes from the Latin word cella, which means a small room. We have also studied that a cell is the basic structural and functional unit of all living organisms. It is the smallest fundamental unit of life. They are also called the building blocks of life, and the study of cells is called cell biology or cytology. Now, what I would like to tell all the participants is that if you have any queries, please feel free to type your queries in the chat box, and I shall address them at the end of the talk. Next, let's go back to the history of the discovery of a cell. If we go down to the year 1600 and then come back gradually from the past into the present, the story goes like this. Robert Hooke, in the year 1665, he was an English scientist who looked at a thin slice of cork through a compound microscope. He observed many tiny hollow room-like structures which resemble a honeycomb, and therefore he called them cells. Cells actually resembled the rooms in which the monks stayed in a monastery. So from here, he gave this particular term. Then Antony van Leeuwenhoek in the year 1674, with the improved microscope, discovered free living cells in pond water for the first time. In 1831, Robert Brown, he discovered the nucleus. Then in the year 1839, Purkinje, he coined the term protoplasm, which means the living substance or the living matter of a cell. Schleiden and Swan in the year 1839 and 1838 respectively, they proposed the cell theory that all plants and animals are composed of cells. Rudolf Virchow, he expanded the cell theory by saying that all cells arise from pre-existing cells. So, all this is something which you are already familiar about. The history of the discovery of cells, the plant cells, the animal cells. Now, let's take one step ahead. From the discovery of cell, we now come to the next important topic, which is the diversity and commonality of cells. Means they are diverse, yet they are having many things in common. So the diversity is in terms of their shapes, in terms of their sizes. As you can see over here, cells come in an astounding assortment of shapes and sizes. You can see over here that in the first figure, we are seeing the eubacteria cells, figure A. Figure B is showing archaebacteria. C is showing red blood cells, as we can see those red concave shaped cells. D are the large single cell fossilized dinosaur eggs. E is a colonial single cell called green algae. F is neurons. G is the epithelial cells, and H, they are showing the plant cells. So the beauty of nature lies in the fact that cells come in so many different shapes as well as in different sizes. Now, in terms of the diversity in the size of the cells, the first figure is showing the smallest cell which is present in the world, and that is Mycoplasma gallisepticum. The largest cell is that of an ostrich egg. The longest cell is that of a neuron or a nerve cell. The largest cell in the human body is an egg cell. And you'll be surprised to know that the size is 1 millimeter. And the smallest cell of the human body is the sperm cell, which has a size of just 60 micrometer. A nerve cell is so long that it can be over 1 meter long. So you can just imagine the diversity which exists in nature in the size of the cells. Now, why are cells so small? The human body, as I told you all, is composed of 10 trillions of cells. Why so many cells, and why are these cells so small? Now, there is a very beautiful concept behind this. Small cell size results in an increase in the surface area is to volume ratio. Now, what do I mean to say by making this bold statement? If you look at 3 cubes, each cube is having a edge length of 1 millimeter, 2 millimeter, and 4 millimeter cube. So if you just grab a piece of paper and your pens, if you have them near you, and you just calculate the surface area and the volume, and the surface area is to volume ratio. Now, how will you calculate that surface area? You can calculate by applying the formula 6a square, right? Volume is calculated by applying the formula a cube. A is denoting the side length. All this you have already studied, I'm sure, in your class 11, 12. All these formulas are already well known to you all. And finally, if you calculate the surface area is to volume ratio for each, you will be surprised to see that the small cube, which is having an edge length of just 1 millimeter, has the largest surface area to volume ratio of 6. Then the third one, the third cube, which is having a biggest edge length, 4 millimeter, that is showing the smallest surface area is to volume ratio. So you might wonder, so what? So what? If a small cell has a larger surface area to volume ratio, what is the big deal about it? So here is the biological significance of the increase in surface area to volume ratio. Now the volume of a cell determines the amount of metabolic activity which is carried out per unit time. And the surface area determines the amount of substances which enter into it from the outside environment and the amount of waste products which leave from the cell into the outside environment. So the important point over here is that the surface area to volume ratio gets smaller as the cell gets larger. So now let's imagine the opposite case. Okay, the cell is just growing larger and larger beyond a certain limit. In that case, not enough material will be able to cross the membrane fast enough to accommodate the increased cellular volume. Now, when this happens, the cell must divide into smaller cells with favorable surface area as to volume ratio. Otherwise, they will cease to function. So, next. So, I just spoke about the diversity in the size of the cells. There also exists a diversity in the shapes of the cells. As you can see in this figure, red blood cells. The red blood cells are circular and biconcave. Why? Because the shape is such so as to support their passage through narrow capillaries and the transport of oxygen. Next, if you look at the white blood cells, they have a very flexible shape. Okay, they are either amoeboid, means they have kind of pseudopodia, so that it can change their shape. This is because they have to engulf the foreign microorganisms which enter into the body. They are the soldiers of the body. Okay, white blood cells, which impart immunity. Next, if you look at the nerve cell in this figure, it is like a long, thin wire. As we know, you must have seen those electrical wires. The main function is the conduction of electrical pulses, electrical signals. In the same way, these nerve cells in the body is beautifully designed in that fashion so as to conduct the impulse from distant parts of the body, from the brain, and vice versa. Now, if you look at the muscle cells, the muscle cells, the striated muscle fiber over here, it is long, spindle-shaped and contractile. Why is it shaped in that fashion? So as to pull or squeeze different parts of the body. And finally, if you look at the figure, the guard cells. The guard cells, you can see the stomata, right? They are bean-shaped. Why? So that they can open or close the pores. So all the different cells in the body, they have been assigned shapes in such a fashion that they perform the function which they are supposed to do. So that is the beauty of nature. So that was all about the diversity of the cells. What about the commonality of the cells? What is common? Okay, all cells definitely share certain structural features and carry out many complicated processes in basically the same way. If you look at this figure, we have a prokaryotic cell and a eukaryotic cell. Okay, so that is the broad classification of all the cells in the world. Prokaryotic cells, they lack a true nucleus, whereas a eukaryotic cell has a true nucleus. Prokaryon, karyon means nucleus, and eukaryon, U means true nucleus. So that is the commonality of all the cells, that they are broadly classified into either prokaryotic or into eukaryotic. Next, so what is the difference between a prokaryotic and a eukaryotic cell? Well, this again might sound a bit repetitive, but we are just brushing up the basics right now. So a prokaryotic cell, we are actually referring to those cells which are bacterial, archaebacterial or eubacterial. Okay, and they're very simple. They're less complex. But the eukaryotic cells, they're found in human beings, plants, animals, insects, and so on. A eukaryotic cell has a well-defined true nucleus, whereas a prokaryotic cell lacks that nucleus. The DNA of eukaryotic cells have linear strands, whereas the DNA of prokaryotic cells is circular in nature. So, next, coming to the difference between a plant cell and an animal cell, this again is a recapitulation. We are all aware of the fact that plant cells are larger in size compared to an animal cell, which is much smaller in size. Then, in a plant cell, the nucleus is located at the side, whereas in animal cell, it is located in the center. Then, a plant cell consists of a cell wall, which is absent in an animal cell. It consists of chloroplast, which is responsible for performing photosynthesis. It has a big vacuole. So, next, before I moved into, as I started my presentation saying that a cell is the basic structural and functional unit of life. So, before going into the function of a cell, let us just have a look at the structure of the cell. Okay, so just take a minute and try to recapitulate what you have already studied in your class 12 level. Cell is containing several organelles, and these organelles, different organelles, have different functions or roles to perform in the body. Okay, so if you look at this particular slide, the cell's post office, I have given this caption, who am I? Can you just take a minute to think which organelle is called the cell's post office? Which organelle is the brain of the cell? There is an organelle which is called the suicidal bag of the cell. Then the living matter of the cell and the powerhouse of the cell. Take a minute to think. As I now move on with my presentation, I will gradually unravel to you these different names and their functions. So, from here, we move on into the structure of a cell. This is the introduction to the cell structure. They consist of cell boundaries, which can be the cell wall and the cell membrane, and they consist of the protoplasm, which is the living matter of the cell. The protoplasm consists of the cytoplasm and the nucleus, and the cytoplasm in turn, it consists of the cellular organelles, vacuoles, and cytoplasmic inclusions. Now, these cell organelles, they can be either membrane bound or they can be free of any membrane. If they are membrane bound, it can be either a single membrane bound organelle, which is in the case of endoplasmic reticulum, Golgi apparatus, lysosome, and if they are double membrane bound, they are comprising of mitochondria or the plastid. Now, if they are not containing any membrane, what kind of organelles are they? Ribosome and the centrosome. So these points you need to remember. So coming to the first important component, the cell wall. You can see in this slide, there is a house in the center, which is surrounded by a fence. It is like a fort. Okay, so the cell wall is like a fort. This house in the center, I have named that as the nucleus. Okay, and the cell wall is the fence surrounding this fort. It protects the organelles inside the cell and also provides the strength and support to the cell. So, what about its location? Where is the cell wall found? I am sure you all know it is a tough, rigid layer which is located outside the cell membrane. It is found, as you can see, in the plant cell, but it is absent in the animal cell. The composition of the cell wall in the plant cell is primarily cellulose, and in case of bacteria, it is peptidoglycan and lignin in case of fungi. So what is the function? I just mentioned it provides the strength, the rigidity to the cell, and it protects the cell against any kind of mechanical stress and infection. Next, the cell membrane. Now, this is a very interesting feature. The cell membrane, it consists of a phospholipid bilayer. You must have heard of several models, the Singer-Nicholson model, but as we don't have that much of time, I would just give you all a gist of the location of the cell membrane. It is present in all cells, animal or plant cells. It's present everywhere. Okay, in case of plant cells, it is just present beneath the cell. Next, what is the composition? It is composed of the phospholipid bilayer, which can be embedded with proteins and a small amount of carbohydrates. If you look at this figure, you can see that the phospholipid bilayer, it consists of an intracellular and extracellular part. It consists of hydrophilic head, means water-loving. Hydrophilic means water-loving. And it consists of hydrophobic tails, means water-hating. The hydrophobic tails are present in the center and the hydrophilic heads are exposed to the aqueous environment. So what is the function of this membrane? It is a semi-permeable membrane which allows selected molecules to move into and out of the cell, okay, to diffuse across the membrane. So just a brief overview of the two types of transports which take place via the membrane: active transport and passive transport. Okay, as you can see, in case of passive transport, again, you have two types of diffusion. One is simple and the other is facilitated. Okay, in simple diffusion, the molecules are moving from a region of their higher concentration to a region of their lower concentration directly through the membrane. In case of facilitated diffusion, they are again moving from a region of their higher concentration to a region of their lower concentration, but they require a carrier or a transporter protein for this function. Now, coming to the active transport, the molecules are moving against the concentration gradient, means from a lower to a higher concentration. As you can see, this diamond-shaped molecules, you can see only 2 over here and we have 5 inside the cell. So if it has to move from their lower to their higher concentration, they will obviously require some energy in the form of ATP. Okay, so this is the main function of the cell membrane. And now, the term which I had used in that who am I slide, the living matter of the cell. Who am I? It is the protoplasm. The living part of the cell is the protoplasm, which is composed of the cytoplasm plus the nucleus. Okay, the cytoplasm is a jelly-like substance which is enclosed by the membrane that contains the organelles to carry out specific functions, and it also provides a medium for various chemical reactions. Whereas the nucleus, it contains a hereditary material, that is the genetic material of the cell. So this figure shows you that the protoplasm is nothing but the sum total of the cytoplasm plus the nucleus. So next, coming to the brain of the cell. The next term which I had asked is the brain of the cell. So it is the nucleus which is known as the brain of the cell, right? As you look at the structure of the nucleus over here, it consists of tiny nuclear pores. Okay, as you can see over here, these nuclear pores are nothing but perforations in the double-layered nuclear envelope, which regulate the passage of molecules between the nucleus and the cytoplasm. Inside, enclosed within the nuclear envelope, is the nucleoplasm, which is the fluid material or the suspension medium. Then in the center, you have the nucleolus, which is a membraneless organelle and it manufactures the ribosomes. Then you also have the chromatin, which is a complex of DNA protein, and when the cell prepares to divide, the chromatin condenses into chromosomes. Next, the term which I had used in the former slide, the powerhouse of the cell. So, which is the powerhouse of the cell? Which organelle? It is the mitochondria. Why is it so called? Because it produces energy in the form of ATP. This also has a wonderful structure. It consists of an outer membrane and an inner membrane, which is thrown into folds known as cristae. And you even have an intermembrane space and an intracellular matrix. The outer membrane is freely permeable to nutrients, ATP, and other molecules. And the cristae, okay, the folds inside of the inner membrane aid in the production of ATP. So, as it is the powerhouse of the cell, production of energy in the form of ATP, okay, is the most important function of mitochondria, and it is also the site of oxidative phosphorylation. Next, coming to the next organelle, plastids. The three types which exist are chloroplast, chromoplast, and the leucoplast. Chloroplast contains chlorophyll, which is a green pigment that absorbs sunlight for performing photosynthesis. It produces and stores glucose. The chromoplast, it contains the carotenoids, which are the red, orange, and yellow pigments, and they're found in flowers and fruits. And leucoplast, as you can see, it contains no pigment, and the function is to store starch. So coming to the chloroplasts, they have a double membrane structure. They have circular naked DNA and 70S ribosome. As you can see, they have inside the chloroplast flattened disc-like structures which are called thylakoids, and they are arranged into stacks called grana. Photosystems are present in the thylakoid membrane and they maximize the light absorption. Stroma is the fluid which is surrounding the grana, which contains the appropriate enzymes for the Calvin cycle. And as you can see, lamella, they connect the grana. So the important function of the chloroplast is photosynthesis. There is a very interesting theory which is known as the endosymbiotic theory to explain the origin of the chloroplast and the mitochondria. According to this endosymbiotic theory, what happened is there were infoldings in the plasma membrane of an ancestral cell, which gave rise to the endomembrane compartments like the nucleus and the endoplasmic reticulum. In a first endosymbiotic event, the ancestral eukaryote consumed the aerobic bacteria, and that evolved into the mitochondria and it gave rise to heterotrophic eukaryote. In the second endosymbiotic event, the event resulted in the consumption of a photosynthetic bacteria which evolved into chloroplasts, and that gave rise to the modern photosynthetic eukaryote. So, endosymbiosis. Endo means to inside, engulf, and symbiotic means a relationship which is mutually beneficial to both the partners. So, it resulted in the production of the chloroplast and the mitochondria. So next, coming to the next organelle, which is the endoplasmic reticulum, it is a network of interconnected membranes called cisternae, which enclose the lumen. At some sites, the endoplasmic reticulum is continuous with the outer nuclear envelope membrane. Now, this endoplasmic reticulum is of two types, RER, which is a rough endoplasmic reticulum, and they have ribosomes on the surface, and the smooth endoplasmic reticulum, which do not have ribosomes studded on the surface. So this is RER and this is the SER without any ribosomes. The function of endoplasmic reticulum is that the cytosolic phase of RER is studded with ribosome and they act as sites for protein synthesis. Whereas the SER, the smooth ones, they lack ribosomes and function in lipid synthesis and the production of steroid hormones and detoxification. Next, I had asked you all in that who am I slide, which organelle is called the cell's post office. As we all know, what is the function? What happens in a post office? The receipt and delivery of various messages and other things. Okay, so the Golgi apparatus also has something similar to perform. The receipt and delivery of certain components of the cell. It is located in the cytoplasm again, and it is located close to the endoplasmic reticulum and the nucleus. Plant cells, Golgi apparatus is called the dictyosome. It consists of membranous components, as you can see, called cisternae and even vesicles. The Golgi complex is polar in nature, means it has a cis phase, which is the receiving department, as you can see over here, the cis phase, and it also has a trans phase, which is the shipping department, the delivery department. Vesicles are originating from the pinching of the cisternae. The primary function is secretion, and it acts as a packaging and transport organ. Okay, just the function which is performed by a post office. And what do they transport? They transport various materials like proteins, polysaccharides out of the cell. Next, we come to the organelle, which I had termed as the suicide bag of the cell. Okay, these are nothing but the lysosomes. What do they digest? They digest food, they digest organelles, they digest cells. Okay, that is why they are called the suicide bag of the cell. Now, what is the structure? They are tiny, regular membrane-shaped bound vesicles which are found in the cytoplasm of both plant as well as animal cells. Since they're helping in digestion, they consist of various hydrolytic enzymes, and they are membranous sacs which are budded off from the Golgi apparatus. So what is the most important function of lysosome? They help in digestion. What are the various enzymes which is present in the lysosome? The nucleases, proteases, carbohydrate digesting enzymes, and so on and so forth. Next, coming to the organelle, the ribosomes. Okay, they are also called ribonucleoproteins. Why? Because they are composed of both protein as well as rRNA. Now, they can be of two types. They can be either free ribosomes, which are suspended freely in the cytoplasm, or they can be bound ribosomes, which are attached to the outside of the endoplasmic reticulum, as in the case of RER, the rough endoplasmic reticulum. As we can see over here, a ribosome consists of two subunits, whether it is a prokaryotic or a eukaryotic ribosome. But there is a difference. Okay, this is a very, very important thing which you should remember, never forget, that a prokaryotic ribosome is 70S. It consists of a large 50S subunit and a small 30S subunit. And a eukaryotic ribosome is 80S in nature, and it consists of a large 60S subunit and a small 40S subunit. And they even differ in the composition of the RNA which is present in the subunits. What is the function of the ribosomes? They play a very important key role in translation, that is in protein synthesis. The genetic message is carried by the mRNA from the nucleus into the cytoplasm, and this message is translated by the ribosomes. Okay, as you all are aware of the protein synthesis pathway, that there is a small growing polypeptide chain and subsequently more amino acids are added to it, and that's resulting in the large polypeptide chain. Next, the centrosomes. The centrosomes are made up of two mutually perpendicular structures which are called the centrioles. The main function is to maintain an equal distribution of chromosomes in daughter cells during cell division, as you can see over here. What about the vacuole, which was present exclusively in the plant cell? It is the empty space. Okay, this is a membrane-bound cavity inside a cell. It stores materials like food, water, nutrients, as well as waste products. And there is a membrane that surrounds this cell waste products and break them down into small pieces. So most on cells depends on how full So, having discussed with you all the various structural aspects of the cell, we now come of the cell. Because the first slide which I showed you all, the textbook definition of a cell. Okay, the cell is a structural and functional unit of life. We have discussed in brief the structural aspect. the various organelles which are involved. Now we move into the functional aspect or the work of the cells. So what is the work of a cell? Cells can build and degrade numerous molecules and structures, numerous macromolecules, proteins, lipids, carbohydrates. They can synthesize many such structures. These processes are driven by ATP, or they can be energy independent processes. And ATP, they are all aware of the fact that it is known as the universal energy currency of the cell. So a cell is nothing but a chemical factory. Okay, just as in a factory, numerous things are manufactured. These tiny little cells, they are also powerhouses. They are also factories which are synthesizing numerous things. They produce an enormous number of complex molecules from simple chemical building blocks. Okay, proteins being synthesized from simple amino acids. You have carbohydrates being synthesized, lipids being synthesized, nucleic acids being synthesized from tiny building blocks called nucleotides. So several complex macromolecules have been synthesized from simple building blocks. And all of the synthetic work is being powered by chemical energy, which is extracted primarily from sugars and fats or by sunlight, means either through respiration or by photosynthesis in plants. And as I again mentioned, it is energy driven. So tapping the energy from the sunlight or by respiration, in case of animal cells, ATP is getting synthesized, and this energy is being utilized for the synthesis of macromolecules, which I just mentioned, in the form of DNA, RNA, proteins, and polysaccharides. There can be synthesis of other cellular constituents like membrane phospholipids. Then this also powers several cellular movements like muscular contraction, relaxation, movement of chromosomes during cell division, mitosis, meiosis. This energy powers transport of molecules, and it results in generation of heat, maintenance of temperature, and it also generates electric potential, which is important for conduction of nerve impulse. So the first function of the cell is to build various molecules and structures. The next important function of a cell is they produce their own extracellular environment and glues. Glues in the sense, the substance which is holding the cells together. So collagen is the single most abundant protein in the animal kingdom, and it is the major component of the extracellular matrix. The cells in the animal tissue are glued by cell adhesion molecules called CAMs, and they are embedded in the surface membranes. Now, if you look at these two structures of the cells, we can see the cytosol of adjacent animal cells is connected by bridges, or which are called gap junctions. And in case of plant cells, two adjacent cells are connected by bridges which are called plasmodesmata. So these structures are allowing the cells to exchange, okay, these gap junctions in animals and plasmodesmata in plants, they allow the exchange of small molecules, including nutrients, signals, facilitating the coordinated functioning of cells in a tissue. Next, cells can also, as just an entire animal or a human being, can move about. In the same way, even a tiny little cell can change its shape, and it can also exhibit movements. Three types of protein filaments organized into networks and bundles form the cytoskeleton. Okay, means it is just as in a human being, the skeleton that is forming the supporting framework of the body, the cytoskeleton forms the supporting framework of the cell. And it consists of intermediate filaments, microtubules, and microfilaments. Okay, they are shown in the figure over here. In actual, they don't exist in these three colors, green, blue, and red. They have been stained in that fashion. That is why they're appearing like that. So the cytoskeleton prevents the plasma membrane of animal cells from just falling limp into a sphere. Okay, it helps in supporting it. And it also functions in cell locomotion and the intracellular transport of vesicles, chromosomes, and macromolecules. Another important function of cells is cell signaling. Okay, they receive and deliver information from outside into the cell. So some of the cells, they have surface receptors. Okay, when the signal is bound to these surface receptors, it results in the conversion of the inactive enzyme to its active form. Another important cell signaling pathway which operates, as we can see, there is a cytosolic receptor. Okay, these receptors can either be membrane bound, as you can see in the first case, or they can be freely located in the cytoplasm. So this receptor is binding to a particular hormone, red color hormone. The moment it binds to the hormone, it translocates into the nucleus. After translocating into the nucleus, it binds to the DNA and triggers the transcription and finally the translation. Okay, from DNA you're getting the RNA by transcription, and then you're getting the protein translation. So cells have this important role in cell signaling. Next, they regulate the gene expression to meet their changing needs. Okay, genes are present on the DNA. As a result of transcription in these cells, you have the primary transcript. Then you have, after transcription, the DNA is getting converted to RNA. From RNA, you get your proteins by translation, and then they undergo certain post-translational modification events. So depending on the needs of the cell, okay, and powered by the cell signaling pathways, they can undergo transcription and translation. Cells, they grow and divide. Okay, shown over here are the various phases of cell division which you all have studied. The G0, G1, S, G2, M phase. So G0 is the resting phase. G1 involves RNA and protein synthesis. S is the DNA replication phase. G2 is the RNA and protein synthesis phase. So as a result of this, why do the cells divide? As I told you all in the beginning, each cell, there's a threshold level of surface area to volume ratio which it needs to maintain. So if it crosses that threshold, they have to divide to meet the needs of the cell. Next, they also determine the gender or the sex of an individual. How? As you can see, a female cell is diploid 2n, and in case of a male, it is 44. As you can see, XY, male cell diploid, and female cell diploid is XX. As a result of meiosis, the division results in from a diploid to a haploid condition. From XX, we have two cells, each containing a single X. And a male, you have one cell containing X and the other cell containing a Y. So depending upon the fertilization which takes place, if X and X unite, it will give you a diploid XX cell and the zygote will be a female. And if X and Y unite, the zygote will be a male. So over here also, the cells determine the sex of an individual. Now, we come to the last slide of my presentation. Having completed this entire journey, the cells are dividing, they are multiplying, they are reproducing, they are performing numerous activities, but at the end of the day, nothing is immortal. All human beings ultimately perish. In the same way, all cells, they complete their journey, they die. The cells die from either an aggravated external assault or they undergo a natural programmed cell death, which is called apoptosis. So here we can see the various hallmarks of apoptosis. Means if a cell undergoes its natural programmed cell death, the cell initially shrinks. This is a journey which it undergoes while it is dying. The cell shrinks, the chromatin condenses, the membrane starts blebbing, means the membrane blebbing means they are just bubbling out. Okay, they are budding out rather. The cell organelles, they disintegrate, the nucleus also disintegrate. Ultimately, the apoptotic bodies form. Macrophages ultimately phagocytose these apoptotic bodies. and there is no inflammatory response over here. Okay, because it is happening inside the cell. It is happening inside. It is not something which is foreign. So this is how a cell ultimately completes its journey from its beginning to its end. We now come to the last slide of this presentation. Thank you very much for your attention.
Yes. So it was a fabulous journey, Professor Calcutawala. I must congratulate you. Without any ticket, without any reservation, we have traveled from the birth of a cell to the death of
That
the
of the
cell.
cell.
Yes, so I mean, it's a fabulous journey. It's a really fabulous journey. So I must thank Professor Calcutawala for giving this very nice and very, I mean, informative lecture. So on behalf of the department, I mean, Professor Calcutta is also part of the department. So actually, she is, I mean, giving thanks to her itself. So, but yes, as I mean, it's the norm that I have to give her a thanks. I cannot, I mean, thank her actually because without Professor Calcutta, I mean, MBBT department is incomplete. So
Thank
she is, yes, she is a very active part of our department. Always, whenever I call Fatima, this is the problem. She is telling that, yes, Rajat, I am solving. So she is
you.
actually a powerhouse of MBBT department. Okay. So,
And Rajata, you are the heart and brain of the department, the soul of the department, rather.
thank you. Thanks a lot. Okay. So, Dr. Kalkatawala, really very nice presentation. I think my students and the participants who are joining, I can see in the live streaming on YouTube channel. There are more than 130 participants are listening to your lecture. So
Okay, thank you.
very nice, very nice. Professor Chattopadhyay, our VC sir, maybe
Has
he is
joined.
facing some problem.
Rajat sir, he has joined. Sir has written in the chat box that sir
Sir
has
has
joined.
joined. Okay,
Yes,
okay, okay. Sir, are you there, sir?
Yes.
Yes, sir. Sir, we all are very glad that you have joined us. And kindly, sir, address my students, I mean, your students. When the mic will be yours, then every one of us will be your students because me, Fatima, Shomshubro, Milan, Atri are also your students, and
yes,
my students are definitely your grand students, sir. So please address all of us, sir.
Thank you, Rajat, and thank you all the faculty members who are present over here. Um, this is a very important event in the MBBT department because we are going to start our new session, academic session, very soon. But this is a preparatory for that. We have the already new students who have enrolled with us, along with the students who are interested to join with us. Now, from the lecture of our head of the department, you came to know about the very exciting departmental activities which we normally have. And from the lecture of Fatima Kalkatawala, you came to know that the quality of the faculties we have with us. Now, this is just a start. But what I can say today that the students who are interested to join in the field of microbiology, biotechnology, related these life science subjects, it may be considered as a completely new era because there are so many different important things happening right now in the whole world because the world is looking right now for cross-disciplinary understanding of knowledge, and being the part of this department you are going to be one of the few in that particular journey. because you know that we in our departments always treat our students not only to learn the subject but also the related subject areas, even the subject which are distantly related to it. Say for example, we are interested that the students can also can get into.
So participants, until sir is again joining, please share your experience for the starting journey with Professor Kalkatawala, and any other information if you want, or any query if you have, then you can mail from what mail ID you have got the invitation for this program. And tomorrow again you are going to have two very interesting lecture. One is from Dr. Sanjay Malik. He will be speaking on cell sorting and how the cell can be differentiated, how the cell can be, separated from one another. And me myself will deliver one lecture on the biomolecules. What are the biomolecules present in our cell and how they can metabolize, how they can produce the energy. These two topics will be delivered tomorrow. So I hope all the participants who are actually listening today's webinar, today's lecture, they have got quite interested, they have got quite enthusiasm from our department. So they will be with us for the coming 4 days. So let's see. And if you have any query or anything related to this webinar or our department, please share with us your views and your thoughts. Always, your thoughts are always welcome for us. Okay. Yes, Fatima.
Yes, Rajat sir, I just had a word with VC sir.
Yes.
He's
Mm-hmm.
saying he's having some network issues. He's got
Okay.
disconnected. He's saying
Okay.
you all can close the Rajat sir. You can close the session on his behalf.
Okay. 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 So, as due to some bad internet connection, our VCSR has got disconnected, and he is not. He's trying, but he is not getting connected with this webinar series. So, on behalf of him, with the due permission to him, on behalf of the all MBBT department, I'm concluding for this first day session, and I'm thanking all my faculty members, and at last I'm thanking all my participants and dear students, in national and international participants. So hoping that tomorrow again we will meet in the same time and with a new journey of this biotechnology and microbiology. So bye for now. Stay safe, stay healthy, be at your home, and keep in touch with us, and take care about your family. So bye for now. Bye today.